Well, this is called the Wolfson Field Lab. It was set up with funding from the Wolfson Foundation and the Royal Society. And it's about measuring greenhouse gas emissions from soils and particularly turnover of soil carbon. And that's of interest because there's a lot of interest in trying to sequester more carbon in soils globally. But the processes involved are actually quite complicated. This lab is about developing better understanding and models to answer those sorts of questions. These are called lysometers. There are 24 of them here. A lysometer is a block of soil which is isolated from the surroundings so that water flowing in doesn't flow out of the sides, basically. So we can measure what comes in at the top and what leaves at the top and what leaves at the bottom. There are two soil types, and we went out and collected these large cores of soil. They're a metre deep and 80 centimetres across, and we wanted to collect them without disturbing the soil, so with their natural structure. And we went to two sites, one near here which is an acid sand, so quite free draining, the water runs through it. And then to contrast with that, we wanted a less free draining soil. And we had to go all the way to Warwickshire to find the right one. And the clever stuff is on top of them, gas flux chambers, so lids that close over the soil surface, and they're connected to a gas line which pumps air around into the chamber once the lid is closed, and out the other side, and it goes around a loop into a lab where we have instruments for measuring the gases that have come off the soil. So what's happening now is this lid is closing, and it will make a gas-tight seal on the chamber here. It closes fast, and then the last little bit is slow. We don't want a big whoomph of pressure which will displace gases. So the last bit is slow, and there's actually a little pressure release valve here which closes after the main lid. And there's a little fan in there which is circulating the air. There's an inlet pipe here and an outlet pipe here which goes around a loop into the lab where there are instruments for measuring the gases. The lid closes, the air is circulated. It takes maybe a minute for the air in the chamber to equilibrate with the small volume of air in this loop. There's something like 100 litres of air in the chamber and 1 litre in the loop. And once that's happened, we start recording the accumulation of, in this case, carbon dioxide, which is being emitted both from the soil and the plants. Now, currently the lid is transparent, which means whilst the lid is closing, the grass is continuing to fix CO2 in photosynthesis. So it's actually withdrawing CO2 from the chamber. But what we're really interested in is the release of CO2 from the plants in the soil. So to measure that, we cover this lid with we have another foil lid so that the chamber is blacked up when it's closed. That means photosynthesis stops, and it stops pretty quickly. And then we're just measuring what's coming out of the soil and the plants. And from the isotope composition of the CO2, as I was saying, we can separate those two out. So that's really the main point of it. At different depths in the soil underneath, we have moisture and temperature sensors. So 5, 10, 15 and 50 centimetres, I think. And then we collect water that's running through to the bottom, metre deep, and any carbon or other things that are accumulated in that. And that gets drained into a collection point in the manhole here. The manhole also contains all the valves and electronics for controlling the gas flow and the measurement system. If it takes 15 minutes to close and open and then move on to the next one, that means each of the 24 can be sampled 4 times in 24 hours. But we don't want to do it in the same sequence. We want to have each one sampled at a different time each day and so on. So the computer in there every morning sets a random sequence for that day and then goes around. And by doing that more or less continuously, we get nice, more or less continuous data sets for making mathematical models. The research here is about understanding the fundamental processes that influence how rapidly carbon is turned over, how fast it's being released from plant roots, and then the effect of that new carbon on the carbon that's already in the soil. Well, we're just coming to the end of a PhD project. The next project we are planning to set up is actually about inorganic carbon in the soil as well as this organic carbon I've been talking about. And the idea here is that it may be possible to fix carbon as carbonates in the soil by adding the right minerals. It's a unique lab. I don't think anybody else has got one quite like this. We're fortunate to get a nice big chunk of money to do it, and I'm also fortunate to have some very good people to work with set up. So particularly the hydraulics that open and close these gas chambers. The other sort of interesting side of it is the gas measurement side, and particularly this aspect of using isotopes. So we don't just measure the gases that come off, we measure the isotopic composition. And that also, we were very fortunate to have some good experts to collaborate with from a mass spectrometry company. And they were excited to get involved with a sort of weird project like this.